Okay, so we can get started. And what we're going to do today is we're going to continue that story about synapses we started last time. And I got a little out of phase between the first section and the second section, so trying to bring them back into phase. And what we're going to do is we're going to start with this idea of the structure and function of the synapse. It's going to be the general topic for today. And I want to walk you through some of the history of how these things were discovered. There are some names here. Never remember these names. I'll never ask you about names. But there's some concepts that these guys developed that are really important. First was Sherrington. Sherrington was the guy who named the synapse. He was a scientist in the late 1800s, early 1900s, and he recognized if the neuron doctrine were true, and he attended a lot of Santiago Ramon y Cajal's lectures, and he recognized that there was something to all that the neuron was the basic fundamental unit, structural and functional unit of the nervous system. But he recognized that if you're going to have that arrangement, that there had to be some kind of communication from one neuron to the next. He didn't know how that was going to happen, but he imagined two neurons coming close together, and so Sherrington developed the concept of the synapse. He named the synapse. It comes from the Greek. It means to clasp or hold. Then there was another contemporaneous scientist with Sherrington, a guy named Langley. Langley was a pharmacologist. Where Sherrington was interested in the physiology, Langley was interested in how molecules interacted, particularly with the autonomic nervous system. The story I'm going to tell you today is how Langley discovered something known as the receptive substance. He described how there must be some kind of chemical receptive substance on muscle fibers. And then I'll talk about Otto Loewi. Otto Loewi and Dale were the ones who really described chemical synaptic transmission. Up to this time, and even for decades afterward, there was an argument as to whether neurotransmission occurred through electrical connectivity, so-called sparks, or through chemical synaptic transmission, so-called SUPs. And we're going to see the SUP on, and much of that is due to Otto Loewi's work in the late 19-teens, early 1920s. And then after the chemical idea of synaptic transmission was established, it took another, probably 40 years before people had tools to actually look at synapses. The major tool that came on board in the mid-50s to mid-60s was something called electron microscopy. And in electron microscopy, they now had the ability to look at synapses. They could look at synapses in the brain, they could look at synapses at the neuromuscular synapse, and they could see the detail. Electron microscopy increased the resolution of microscopy by at least 100-fold. And so now you began to be able to see individual synaptic structures. And this is really what cemented the idea of chemical synaptic transmission, particularly the idea that neurons were separate structures and they never fused one with the other. There are rare cases of gap junctions, but by and large, we're going to see that this idea of chemical synaptic transmission comes down to visualizing the synapse. And when they visualized the synapse, they found the presynaptic cell, the output region of the neuron, contained thousands and thousands of these tiny bubbles called synaptic vesicles. They then realized that some of these tiny bubbles would fuse the plasma membrane and release neurotransmitter, but this didn't occur all over the membrane terminal, or the axon terminal. It occurred in particular regions called active zones. And what characterized the active zones is, first, they were clustered around voltage-gated calcium channels, and secondly, the vesicles were very, very close to the plasma membrane. They were said to be docked at the plasma membrane, and we'll draw this out. At this point, they recognized that calcium was necessary to cause neurotransmitter release. They saw that vesicle exocytosis was likely the cause of neurotransmitter release. It required calcium, but they didn't know the molecular mechanisms. And that came about by describing something called the snare hypothesis. And the snare hypothesis was actually awarded the Nobel Prize in 2012. The snare hypothesis shows that there are these proteins, some of which are in the vesicle, and some of which are in the plasma membrane, that work together. They'll actually literally wind together to smush the synaptic vesicle membrane into the plasma membrane. And we'll talk a little bit about that. But once the vesicle releases its neurotransmitter, once that phospholipid bilayer of the vesicle has been fused with the phospholipid bilayer of the axon terminal, that neurotransmitter is released, but it has to do something. And that will be the next step. Let's then think about what's going on the postsynaptic cell. So much of today's discussion is going to be on the presynaptic cell, but then we'll talk a little bit about the postsynaptic cell, where we see the neurotransmitter binds to specific receptors in that membrane. These receptors are known as ligand-gated ionotropic channels. There's a certain amount of meaning in that. Ligand-gated, it tells you that there's a ligand. In this case, it's going to be the neurotransmitter. Ligand means to bind or to tie, so the ligand binds to the receptor. When the ligand binds to the receptor, that causes a conformational change in the receptor, and a channel opens and sodium flows in for our story today. The influx of sodium produces a very tiny depolarization. This depolarization gets a name. It's called an excitatory postsynaptic potential. And it's the result of opening, probably a few hundred at least, of these ligand-gated ionotropic channels. But the EPSP generated at an individual synaptic site is quite small, probably well less than half a millivolt. And we're going to see that the synapses are at some distance from the trigger zone. We talked about a trigger zone yesterday in sensory neurons. We're going to talk about a trigger zone again, maybe today, probably Monday. A trigger zone at the site where the cell body meets the conductor region. At the portion of the membrane where the cell body meets the conductor region, there's a clustering of voltage-gated channels. That's called, again, a trigger zone. The decision-making part of this is we have an input region which is receiving neurotransmitter, opening ligand-gated channels, generating excitatory postsynaptic potentials. Each of these excitatory postsynaptic potentials is way, way too small to bring the membrane to threshold at the trigger zone. So what it means is we have to add together, we have to summate maybe 20, maybe 100, maybe more of these individual EPSPs at the trigger zone for that decision-making event to say, yes, fire an action potential. So each EPSP is small. It needs to summate at the trigger zone, but in addition, it's got to travel. It has to propagate along the membrane of the dendrites, the membrane of the neuronal cell body. One of the characteristics of the membrane of the neuronal cell body and the dendrites is they lack voltage-gated channels, for the most part. So we can't regenerate this potential. It's all passive membrane. And remember, passive membrane contains leak channels, so what's going to happen is that these EPSPs, as they propagate along the input region, the membrane, which is the input region, they're going to get smaller and smaller and smaller and smaller. So that means it's going to take, the smaller and smaller they get, the more of them it's going to take to reach threshold. And then finally, what we'll see is that we can, by changing, by modifying the membrane characteristics of the input region of the cell, we can modify the amplitude of these potentials. Now, I'm going to exaggerate this a bit. There are more aspects to these equations, so you look at them. But what I want you to know is that if we change the membrane resistance, we have a big effect on the amplitude of the EPSPs. And that's shown by this equation called Ohm's law. Ohm's law says the voltage difference, the voltage change, is equal to the current, the number of ions, times the membrane resistance. We're going to talk about the membrane resistance in terms of how many leak channels there are. The more leak channels there are, the lower the resistance. The fewer leak channels there are, the fewer pathways we have for the ions to leave the cell, the higher the resistance. So we'll see that we can vary this number, R. We're going to fix the number of ions that move at individual synapses. And then that will show us that we have a variation in voltage. I don't know if there's going to be time today, but we'll try to walk through this just with the potassium leak channels. And then on Monday, we'll get around to showing how we can really change membrane resistance using inhibition. But let's start here with the idea of what is a synapse. And again, this term synapse was coined by a guy named Sherrington. And Sherrington said that if the neuron doctrine is correct, there must be sites where neurons communicate one with the other. And he said that neurons communicate at sites where they clasp or hold one another. So he envisioned sites where neurons would come very close to one another and somehow they would hold on to each other. And from that vision, he coined this term synapse, which comes from the Greek meaning to clasp or hold. So from this, in the early part of the 20th century, about 1905, there was general recognition that there must be sites where neurons talk to one another. But that gave rise to two possible ways of it working. And I sort of generalized this into soups and sparks. The soups thinking was that between neurons used chemicals. We're going to call this now we know these are neurotransmitters. The Sparks people were really almost as though they were still in the reticular world. And they said, now, communication, neuron to neuron communication, is through electrical signaling. They imagine a direct electrical signal going from one cell to the next, hence this lost out. Probably is some, in terms of gap junctions. We have gap junctions in the central nervous system. It's probably a little bit of it. But by and large, 99.9% of synaptic transmission in the peripheral and the central nervous system is going to be carried out by soups. But this opened up a whole series of questions. And one of the first approaches was carried out by a guy named John Langley. And John Langley was a pharmacologist. He was interested in how chemicals, particularly plant alkaloids, would interact with biological systems. He was particularly interested in the autonomic nervous system. But he also was interested in muscle. He recognized that if he had nicotine in a whole animal, it caused muscle contraction. So he wanted to know if he could activate individual muscle cells using nicotine. So he did this experiment. He killed a mouse and he dissected out an individual muscle fiber. So this is meant to be an individual muscle cell. He could see the site where the neuron came down close to the muscle. And this is the motor neuron forming the neuromuscular. Junction. In these early days, it was sometimes known as an end plate. So he could see that. The next thing he did is he made up a solution of nicotine. The chemist had isolated nicotine from tobacco plants. And so he made up a solution of it. And he knew that if he actually gave this to whole animals, it caused muscle contraction. But he wanted to know how the nicotine interacted with the muscle. So he did an experiment where he took a camel's hair brush and he reduced the camel's hair brush down to a single fiber. He then dipped that single fiber into the nicotine solution. Then, watching under the microscope, he took the single hair of the camel's hair brush and he'd touch it to the muscle fiber. And as he was watching under the microscope, he would then walk from step to step, moving along the muscle fiber. And he found that nothing happened, and nothing happened, and nothing happened, and then suddenly he got near the site where he could see the nerve terminal. And at this point, the nicotine caused muscle contraction. This gave rise to the concept of a receptive substance. So at this point, what did they know? Well, they knew that there was some kind of receptive substance that would respond to nicotine and cause the muscle to contract. That's fine, but it doesn't really sort out the question of soups and sparks, because nicotine is a plant alkaloid. It's not produced in animals. It's not seen in animals. So it simply tells you that there's some kind of receptive substance for nicotine that causes muscle contraction. It's an important insight, but it doesn't really tell you how the motor neuron's communicating with the muscle fiber. The concept of chemical synaptic transmission was largely sorted out by a guy named Otto Loewi. I'll give you his name. Otto Loewi was trained in Germany, but he was working on this project at a university in Austria called Gans. And he was interested in the autonomic nervous system. He was interested in how the autonomic nervous system would regulate the heart. And his preferred model was frog hearts. And so he knew what he could do is he could take a beaker with the right kind of solutions. He could take a frog heart and put it into solution. Now hearts, we'll see later when we talk about the cardiovascular system, but hearts are autorhythmic. They beat on their own. They do not have to have direct neuronal control to tell them to beat. They're not like skeletal muscle. So he could dissect out this heart and continue to beat. He also knew, and he was interested in the autonomic nervous system, so he would leave these hearts, he put them out in solution, he would leave attached to the heart a length of vagus nerve. Those of you who had some neuroanatomy, this is also cranial nerve 10. He knew that he could take this long stretch of vagus nerve, which comes all the way from the brainstem down to the heart. He could dissect out the vagus nerve, dissect out the heart, take that bit of vagus nerve, lay it over some electrodes, and when he would shock the vagus nerve through the electrodes, the shock would slow the heart rate. So from this, he said, well, there's some kind of communication between the vagus nerve and the heart that's causing it to slow. But it doesn't solve the issue of soups and sparks. You don't know if it's a direct connection or whether it's a chemical. Now, the next part of the story may be apocryphal, but it's a great story. The story is that Otto Loewi woke up in the middle of the night, and he said, I know how I can solve the problem. And at like 2 in the morning, he went back into the lab and started to kill frogs. And what he did is he did the same experiment again. He isolated the frog heart, had it connected length of vagus nerve, stimulated it. And his insight was that if it's chemical, if it's something the neuron is releasing to affect the muscle, then that chemical would be in the fluid. And then, is he set up a second heart. This heart he didn't stimulate. Not even sure he had the vagus nerve attached. And what he did then is he stimulated this for, some time, maybe hours. Then he took a little bit of fluid from this bath and put it into this bath. And the fluid from the bath where he had stimulated the vagus nerve, just the fluid, caused the heart to slow down. This indicated that there was some kind of chemical being released by the vagus nerve that was slowing the heart rate. In typical German fashion, he named it V-A-G-U-S-S-T-U-F-F, Vagerstoff. Didn't know what it was. But he had a friend who was working at the university, I don't know which one college it was, but it was at Cambridge. And this guy's name was Henry Dale. Henry Dale was a chemist. So it was Henry Dale who showed that the Vagerstoff was in fact acetyl, when you give yourself more room, acetylcholine. So this was the first demonstration that a neuron, could release a chemical, acetylcholine, that could now slow down the heart. That was the first clear demonstration of chemical synaptic transmission. The story goes on from there. Turns out, in 1936, Dale and Otto Loewi share the Nobel Prize. They split the prize evenly. Dale gets half, Loewi gets half. But Loewi's working in Austria. And if you know these dates at all, you see bad things are going to happen. Because 1938 is the Anschluss. Nazi Germany invades Austria. And Otto Loewi was Jewish. And so the Nazis made him a deal. They said, okay, turn over your winnings from the Nobel Prize, and we'll let you go. So he wrote a check, turned over his winnings to the Nazi Party, and they let him go, and he then went and worked for a while with Henry Dale in England. Then later he came to NYU and worked at NYU and made really important contributions in his research at NYU. This was not uncommon. If you look at the history, particularly of cellular neurobiology, there was an enormous exodus of young scientists out of Central Europe at that time. Most of them went to England. Some of them came to the States. And these young scientists then went on to develop most of the foundations of cellular neurobiology. In fact, there's a famous physical chemist, a guy named A.V. Hill. He's the guy for whom the Hill coefficient's named. Those of you who've had some biochemistry will recognize that. And A.V. Hill then gathered up many of these expatriates and put them in his lab. And one of those expatriates is a guy named Bernard Katz, who was very instrumental in describing what we're going to talk about in a few minutes, the release of neurotransmitter of molecules in synaptic vesicles. He won the Nobel Prize for that. But A.V. Hill used to joke. He said, I always keep a statue of Hitler in my desk because Hitler sent me so many wonderful scientists. And I think it's a cautionary note. When you have these upheavals, when you have these upheavals like we had in the 30s in Germany, maybe like we're having this very day in Central Europe, that causes an exodus of people. They leave, they have to get out. And if you take these people in, they can often be some of the best contributors to your society. And in this particular case, it was Otto Loewi and Bernard Katz and a whole bunch of other people. And so be aware when you see these exodus, when you see refugees, imagine how they can help your society. Don't be afraid of them, because we have this long history of people coming here and developing new techniques, new ideas, fertilizing new fields of research, from these particular refugees. And I think we're going to see a bunch more here in the next few weeks. So with that story, let's now go on from, this work was done late 19-teens, early 1920s, between 1919 and 1921. Then it took another 40 years before there was the development of another really important tool. And the next important tool was the electron microscope. And the electron microscope allowed for the research into the structure of biological systems, which was at least 100, maybe 1,000 times more powerful than the old electron microscopes. And so with the advent of electron microscopy, they could finally see individual synapses. And with electron microscopy, they could finally see the output region of individual neurons. So this is going to be the output region. I'm going to give it another name. I'm going to call it the presynaptic element. Then here in blue, I'm going to show the input region of a neighboring neuron. I'm also going to rename this. I'm going to call this the post-synaptic element. Sometimes you'll see this referred to as the pre- and postsynaptic cell. And one of the insights, one of the important contributions to the neuron doctrine, is that there was always a space between these two. And they could see the space. It was, in the neighborhood of about, 50 nanometers. That space was always there, and they gave it a name. They called it the synaptic cleft. Sometimes you'll see this referred to as the synaptic gap. It's the space between the presynaptic cell and the postsynaptic cell. The other thing that they recognized, what they spotted, was whenever they looked at these output regions at a synapse, this region was just chock full of tiny called synaptic vesicles. Bernard Katz looked at these and said, man, I'll bet each one of these vesicles contains neurotransmitters, and I'll bet what's happening is in some way this vesicle is fusing with the plasma membrane via exocytosis to release neurotransmitter. Well, again, it's a lovely hypothesis, and it took years and years and years of work to actually show this was the case. And after about, probably 20, 30 years work, 25 years work, it was pretty clear the synaptic vesicles were fusing with the plasma membrane. It was pretty clear that they were releasing in the neighborhood of about, 5,000 molecules of neurotransmitter. It was also pretty clear from physiological experiments that to have this exocytotic event occur, have in the postsynaptic, they didn't have the presynaptic, these voltage-gated calcium channels. So what was known is that you needed calcium on the outside. You needed to have calcium in the extracellular space. It was known that the voltage-gated channels which are opened by charge, would allow the calcium to come in. It was known that that calcium in some way would cause the phospholipid bilayer of the vesicle to fuse with the phospholipid bilayer of the plasma membrane. The morphologists, the anatomists, and the physiologists were happy with this. Fine, we need calcium, synaptic vesicle fuses the membrane, no problem. The issue arose when the physical chemists began to think about this. And the physical chemists said, you guys have a problem. It can't be that simple. Because recognize that the phospholipid bilayer is going to have positive charge on it. All those phospholipids have positive charge. And then there are another round of phospholipids here in the plasma membrane, and they all have charge. And they're all similarly positively charged. What's more is there's a water layer between those two. So now imagine we have the vesicle, and we bring it at molecular distances. We bring it closer and closer to the plasma membrane. Once it gets within a certain region of the plasma membrane, probably a nanometer or so, then those repulsive forces push the two apart. So they had to hypothesize at this point some kind of interaction of molecules that would overcome that energy barrier, the energy barrier of the charges pushing the vesicle away from the plasma membrane. And that started a whole new round of experiments and basically molecular biology studies to sort this out. And it brought us to something known as the snare hypothesis. Of neurotransmitter, well, let me do it this way, of exocytosis. I find it interesting that the snare hypothesis was awarded the Nobel Prize. The thinking behind the snare hypothesis and the experiments was awarded the Nobel Prize in 2012. And one of the reasons I find it interesting is that there were 3 individuals who shared the Nobel Prize that year. And each of them made contributions from a different angle. I won't give you the names. One of them was called Randy Sheckman, and he was interested in secretionins. In yeast. He knew that these yeast cells would secrete molecules, and he would mutagenize the yeast cells to see if he could change the mechanism, to change the secretions. And he found a whole series of them, a whole series of mutations called sec mutants. So he was responsible for the sec mutants. So that told him there were proteins involved in secretion. There was another group. This guy's name was James Rothman, and Rothman was working at Princeton, and he was interested in vesicle trafficking. This is the vesicle trafficking we talked about earlier, where vesicles bud off from the endoplasmic reticulum, they move to the Golgi apparatus, they fuse the Golgi apparatus. So again, we have this conundrum of how the vesicles fuse to the plasma membrane. And he found proteins necessary for vesicle traffic. The third guy is a guy named Tom Südhoff. And Tom Südhoff was working, he's now at Stanford. At the time, he was working at University of Texas, Dallas branch, medical branch. And what he was working on is brains and synapses, chemical synapses. What these 3 lines of research brought together was the idea of the snare hypothesis. And with the culmination of the snare hypothesis, we can envision what's going on at the synapse. We can envision how calcium is regulating the fusion of the synaptic vesicle with the plasma membrane. We can envision at a molecular level the relationship between these molecules and how they overcome that energy barrier. And that's what we're going to talk about here. In the snare hypothesis, we see that, as before, we have the plasma membrane. We see that there is a vesicles. And these are vesicles docked at the presynaptic membrane. These vesicles are not docked at the membrane randomly, helter-skelter. They are docked at the membrane right next door to the voltage-gated calcium channels. Taken together, the docked vesicles and the voltage-gated channels produce something known as the active zone. The active zone is the site where we're going to get vesicular exocytosis. Now, if we blow this up, what they found when they isolated these proteins is they found that there were a series of these proteins. One set of the proteins were in the synaptic vesicle. I'm going to show these in orange. They come across the membrane. They're transmembrane proteins that come out into the cytoplasm. I'm going to give this protein a name. This is called VAMP. Vesicle-associated membrane protein. This is the term I'll use. If you're doing reading on this, you'll also see this referred to as synaptobrevin. Synaptobrevin is the term that was coined by Tom Suthoff. I prefer VAMP, although it's not quite accurate in its description. I prefer VAMP because it tells us where it is. It's vesicle-associated membrane protein. It's in the vesicle. It's a transmembrane protein in the vesicle. There's another protein in here. It's a transmembrane protein. This protein is named syntaxin. And syntaxin is a transmembrane protein, abbreviated here, TMP. It's a transmembrane protein in the axon terminal. So, and these two are more or less facing each other. And then there's a third protein. This third protein is not a transmembrane protein. It's associated with the membrane because it's tied into the membrane by fatty acids. Don't worry about it. Just, it's not a transmembrane protein. It's different from VAMP and syntaxin. But all of these form a kind of aggregate. And so they form an aggregate which seems to be stable. Then off to the side here, as we showed over here, are the voltage-gated calcium channels. The action potential arrives at the terminal. It provides positive charge. The positive charge opens the voltage-gated calcium channels. The calcium is in the extracellular space. Calcium in the outside of the cell is 2 to 3 millimolar. It's like 0.1 nanomolar inside the cell. There's a tremendous gradient. So we open the gates. Calcium comes in. The final character in this play is another transmembrane protein in the vesicle. I'll show it as a kind of tag. This protein is called synaptotagmin. The synaptotagmin tegment is the calcium sensor. So what's happening here is a little unclear. We don't know exactly. There are two models. One model says calcium comes in, binds to the synaptic tegment, the calcium sensor. When it binds the calcium sensor, the calcium being bound to the synaptic tegment in some way tells these proteins, the syntaxin, the SNAP25, and the VAMP, to wind together to make a supercoil. These squiggles out here are intended to show that this is alpha helical. And so they associate with one another to be stable. And they can stably sit in that position. What makes the system unstable, and this is again we don't quite understand, calcium comes in, binds the synaptic tegment, and at that point, one way or another, these alpha helices all wind together. The way I envision this is if when you were a kid and you had one of those balsa wood airplanes and it was driven by a propeller to a rubber band, and you'd wind it up and you'd watch the rubber band as it wound itself around, as it wound around itself over and over again, it formed these knots. That's what I imagine is going on here. That these alpha helical regions are winding around one another. And by winding around one another, they're shortening the distance between the membrane of the vesicle and the plasma membrane. And there's enough energy in this winding together of the alpha helices that it can overcome that energy barrier of the charge on the phospholipids. And you will now literally smush the phospholipids of the vesicle into phospholipids. And that causes fibrosis. The debate right now, as far as I know, is about these synaptic tegment molecules. And this is really a detail. We know the synaptic tegment is the calcium sensor. We know in some way it's causing these alpha helices to wind together. But the two models are, one is it binds calcium, and that's instructive. It tells those alpha helices to wind together. So that's the instructive model. The other model is a permissive model. It says that the synaptic tegment is constantly holding back the winding together of these alpha helical structures. It's holding that at bay. And then when the calcium binds, it loses its ability to hold that winding together at bay, and the default pathway is to wind them together and smush the vesicular membrane into the plasma membrane. There is additional evidence that it looks like these vesicles may be sort of breathing in and out, in and out, in and out. And only when the calcium comes in, when the vesicle's in the right position, that these can wind together and overcome the energy barrier to pull the vesicle into the plasma membrane. Yes?
How does the synapse decide the neuron decide which vesicle gets exocytosed?
It's a great question. We'll have an individual synapse, we may have a few hundred of these. There may be several thousand at the neuromuscular synapse. How is the decision made to say, we're going to let this vesicle fuse and not its neighbor? We don't know entirely. Some models say it depends on the number of calcium channels here. It's probably not right. The more common model, the one that I think that Jack McMahon has been putting forward more recently, is that calcium comes through these channels, but the decision to get the vesicle to fuse has to do with the microsecond position of the vesicles with respect to the membrane. He's shown, doing lots and lots of microscopy, that these vesicles come close to the membrane, then they come back. They're breathing in and out. And his hypothesis is it's only those vesicles that are just about at the membrane that when the calcium comes in, they fuse. And that's how the choice is made, a subset. But you're absolutely right. We only release a fraction of the vesicles at all of the active zones. And Jack McMahon has been working on that for 40 years. Does that answer your question?
Yes, but I have a follow-up with that.
Okay.
So do the vesicles all contain just an assortment of different neurotransmitters, or is it specific?
Okay, great question. The question is, do these vesicles contain an assortment of neurotransmitters, or they all contain one type? For years and years and years, there was something called Dale's principle, same Henry Dale. And Henry Dale said that each neuron would only have one type of neurotransmitter. We later found that's not true. We find there's transitions in some neurons. They'll change over from one kind of transmitter to the next. In other cases, we have peptide neurotransmitters that seem to be in neurons along with these other small molecule neurotransmitters like glutamate. The most recent data here say that in the cytoplasm we have enzymes. And these enzymes form the neurotransmitter. Some neurons will contain enzymes for making acetylcholine. Some neurons will contain enzymes for making GABA, others for making glutamate. And then the cytoplasmic proteins make these cytoplasmic enzymes make the neurotransmitter, and then there are pumps on the membrane of the vesicles. And they're an exchange pump where they kick a hydrogen ion out and they move a neurotransmitter molecule into the vesicle. That's how we load the vesicles. But this was a confusion for a long time too. We want to put the neurotransmitter into the vesicles because that's where it's functional. But we kept finding high concentrations of neurotransmitter in the cytoplasm, several millimolar at times. And the answer is we keep high concentrations of neurotransmitter in the cytoplasm, and then we selectively pump those neurotransmitters into the vesicle using these exchange pumps.
Thank you.
Yes?
Can you clarify how the secretion mutations of the snare hypothesis play into this?
So what's the connection between the secretion mutants and the snare hypothesis? Well, when they started looking and sequencing the proteins, they found that the family of proteins that Randy Sheckman had found that were affecting secretion, the amino acid sequences in those proteins were very similar to the SNAP25 and syntaxin and VAMP. So that brought together all the way from yeast to complicated metazoans this idea that there are common proteins. And those kinds of proteins are also, they're not exactly the same proteins, they're slightly different genes, but they're the same gene family. And those proteins are involved in secretion in yeast, they're involved in getting vesicles to fuse to the plasma membrane of the, getting vesicles to fuse to the membranes of the Golgi apparatus, and they're involved in the fusion of vesicles with the plasma membrane in neurotransmitter release. So that's how it all ties together. Yes?
Through all this, I still don't understand why we call these soups. Like there's sparks, and that was electrochemicals. Where's the soup here? Is this
The soup is the soup are the chemicals in the soup, and the soup is the extracellular space. We put chemicals into the extracellular space that open them. Anything else before I spend the last 3 minutes on the postsynaptic membrane? Let's go to the back panel. So we've managed to release the neurotransmitter. Where I want to leave you is the vesicle is fused to the plasma membrane. The neurotransmitter molecules have been released. Over here in orange, this is the postsynaptic membrane. One of the major characteristics of the postsynaptic membrane is that it has thousands upon thousands, or at least hundreds upon hundreds, of these transmembrane proteins called ligand-gated ionotropic channels. That's a mouthful, ligand-gated ionotropic channels, but what it means is that neurotransmitter being released from these vesicles by exocytosis, the neurotransmitter binds to the neurotransmitter receptor. And that this protein that binds the neurotransmitter also is the very same protein that has a channel. And this is going to be a cation channel. It's said to be ionotropic because it passes ions down a concentration gradient. The most common of these is going to be ones which allow sodium to come in. Sodium is positively charged. So this sodium is going to generate a depolarization. that we're going to call an excitatory postsynaptic potential, called a EPSP, excitatory postsynaptic potential. And what we'll do on Monday is we'll talk about how these excitatory postsynaptic potentials are generated by opening these ligand-gated channels, and how these potentials will propagate along the membrane of the input region and summate, add together at the trigger zone